Oh. Hello? How are you? You can't hear me? Can you hear me now?
I think it's stopped.
Let's wait for a few more people to come in. Okay. There he is. Hello?
Good morning.
How are you?
Pretty good.
Good.
Yeah, that's fun. I guess 3 canoeing trips without falling in the water.
Oh, wow. That's pretty good. Where did you go?
What?
Where did you go?
Oh, we went camping a place north of Dauphin, Manitoba.
Okay.
Okay.
Sounds good.
It's from Manitoba.
Okay.
Hello.
Hello. How are you?
Yeah, I'm fine. How are you guys?
Okay.
Exams are almost over. It's 3 or 4 more days.
Oh, good.
After that, I'll be back again on track.
Yeah.
Yeah, so I thought Ujjwal was already updating. Yeah.
That's good.
But I was.
Yeah.
Is Ujjwal there with us right now, or is he? Like,
What was that?
is Ujjwal there with us?
I think so. I think he's having a problem with his audio. I don't know.
My audio is fine, right?
Yeah. Yeah, yours is fine.
Should I start with my update for the week?
Let's wait for a couple more people to show up here.
Yeah.
Alright.
Hello?
Oh,
we can hear it. There we go.
I can hear the audio.
I don't know.
Yeah. So, how have you been, Dick? Do you have any updates?
Last week, I was not able to contribute much on my GSoC project, but pretty good things happened. One of my papers selected in IEEE Big Data and Multimodal Analysis Conference. It's going to be happening around 18 and 19 September in a virtual manner. And also, I got my internship for the next summer. So these are 2 major things that happened last week.
Congratulations then.
Thank you. After that, I have tried to recalibrate myself to the work. I have, as Vinay suggested, compared the Python anywhere in Hero to very thoroughly, and I will share all the results, what is best for what. And I have started working on the C.Delegates embryogenesis segmentation model. which I set up by Pune, and I started working on it, making it more interactive. So which will be done around Thursday, I guess. And it should be our first model which will go live.
That's good. Yeah, congratulations on the internship. You told me about that in Slack. Yeah, so that's good. And I know you've been working hard the last couple of years, so that should hopefully that's what paid off. Yeah. Did you have anything to update us on, Dick?
No.
Oh, okay, I just wanted to give you a chance to.
Well, been working with a fellow in China. I have some no ideas on computer tomography, but I don't think this group wants.
Mayukh, do you have an update?
I have an update. I'll just start with it.
Okay.
Can you guys see it?
Yeah.
Yes.
Yeah. So this week, last week what I worked on was the generative model. What it would do is that it would generate fake images that like fake images of embryos. Like it will generate its own images of embryos. Like we will feed it some noise and it will generate a random image of an embryo. So the way it trains is it's like a min-max game between like two agents. Like first the generator generates a fake image, and then a discriminator tries to discriminate whether or not it's a real image or not. Okay, so there are basically these, like it's trained on images like these. Like these are the kinds of images that it's trained on. So first the generator, like this part would try to generate a model, like generate an image of an embryo, and then this part would try to classify whether or not that image is fake or real. And as the training goes on, both of them get stronger. The generator generates better images, and the discriminator becomes better at identifying a fake, like a generated image versus a real one. As they train, so at the end, the generator becomes better and better and better with further training. So that's what happened. And like the discriminator returns a probability between 0 to 1 on what it thinks is the probability of that generated image being as close to the real image. So the ideal value for the discriminator at the end would be 0.5, because it's perfectly in the middle. It can't decide whether or not it's like whether or not it's real or fake. So the ultimate objective is to confuse the discriminator to get a good generator. So I'll just go into the visualization. That would be better. I'll just share this link. You guys can play it yourself. Wait. Just share this link quick.
All right.
Yeah, on this link, you can just go down and come to this part. Did you get this link? Is it working? You guys just can scroll down and then press this play button, where it will just show how the generator gets better with time. Initially, it just gets better, and this is the end. Like initially, as you can see, like it's just noise. It's not at all trained, and then it starts training, and it gets a basic idea, like of the shape, and it gets to know that some of them are sparse with a less number of cells, and some of them are like really populated with a lot of cells, like in the middle, like one. Like after that, then it slowly like gets smarter and smarter and smarter, and with time it kind of starts to learn the meta features, like things like you can see it, right? I'll just give a closer look on this one. Yeah, can you guys see it now?
Yeah.
Like some of them are like low population, some of them are like high population. So then, and after that, I just generated a few more samples, and like that is it for this notebook. And other than that, what I did was that I extended this, like this is a trained model, right? I saved this trained model, and then I used this model and I extended it into that. Wait, where did it go? I extended and I kept this model as another utility within like this library that I'm working on. So the user can basically generated like it can import generated or generated model like it can just call the class, and then it's as simple as gen image equals to generated.generate. This will return a generated black and white image which the user can work with. Then he or she can show it. And also I added some like another small extension so that it can generate n images of custom size, so that he can generate like 50 or 100 images with the custom size that he wants, like 700 by 500 or something. The native size at which the generator actually generates is 120 by 128, but it can be resized later, right? I can't go much further. I can't like train it on because then the collab GPUs, like the memory runs out. It's very memory, like it takes a lot of time to generate these generative models. So I have to stick with 256 by 256. Like, wait, I'll show you. And then like I just wrote some more scripts which can be used. Like I'll use the scripts in the tutorials where they'll be able to generate like 50 images like in bulk. And then like what I did was that I'll just go back to the README. And then what I did was that I used this deep learning model. Remember it from last week, which could like predict the populations, right?
Yeah.
Yeah, like this one, which I had to clean up my data for this one, right? So I used this model on the generated images, and I got something like this. I'll just zoom on, like I'll just zoom in on it, like I'll just open it on my tab, like. Yeah, can you see it now?
Oh yeah. Yeah.
Yeah. For these images of the embryos, like they're all generated, okay, they're not real. And then I'm feeding those generated images into that deep learning model which I trained long back to find the population. So these are the results. Like, it's kind of like finding something. Now, like the problem with this is that it generates random images. Like it generates, like as you can see on this one, like it generates images with random populations. You can't like specify that I want an embryo from this stage, like from the 1.5 stage, or from the from the 1/4 stage. I can't really determine that. So I am currently trying to work on a model where what happens is that I'll just show that model. I'll just show that as well. Right. I'm currently working on another model that is a conditional GAN. Instead of taking just one input, it will take two inputs. It will take the random noise, and it will also take a bunch of populations of the cells. Wait, I'll just open it up. It will take those populations, and then after taking those populations, it will try to generate an embryo's picture that has those populations exactly, like or approximately those populations. Like if I enter like 11000000, so like it means really low population, right? For all the lineages, it will generate a very sparse kind of an image with a very few cells. But if I give it like 255, 50, 32, something like that, then it will generate a really like an embryo which is almost in its L1 stage, because it's like almost there, right? So now I'm working on this. It's pretty much the same initially. So the interesting part is this part. Like normally, what happens is that I'll just show this part so that you guys kind of get an idea. Like generally, when I'm generating an image, so there are only two conditions. One is I'm basically just taking some noise input and I'm generating an image. So the only argument is input, right?
Yes.
But currently, the model that I'm working on, it has two arguments. Like, can you guys see it?
Yeah.
It's got input and label. Like the generator looks at both of these values. Label means the population. Label means these values, like the population values. Just below these, like you can see it, right?
Yes.
So it takes both of these arguments and tries to generate some kind of an image with that corresponding label. But it's not working right now. The code is basically it's broken. Just don't look at it. It's ugly stuff. Yeah. So that's pretty much my update. I'll start with this after my end semester exams end.
Oh yeah, definitely. Well, that's pretty impressive. Yeah, I saw something about that in the Slack as well. So let's go back. Let me think through this a little bit. You might share your screen again.
Yeah.
You
Sure.
don't want to just talk to nothing. So you have this, yeah. So you have this GAN, this generative adversarial network that you're using to build these embryos, and you're using the data that you got from the data set to generate populations.
Yeah.
So I think that's actually pretty interesting that you have like these data that you're kind of plugging in, and then you're generating these fake embryos, and then you're seeing which ones you're trying to classify them as real and fake. And then
No. I'm trying to classify it as a, like I'm trying to classify them as real or fake as a part. Textbook way of training again. Like you use two models instead of one. One model will generate an image, another model will classify whether or not it's fake. And the target of the generator model is to maximize the loss of the discriminator. Like they're playing against each other. Initially they have zero knowledge, but as training goes on, alternatively they train. Like both of them get mixed one by one, and then they start training. And in the end, we just discard the discriminator because we don't need that, and we take the generator. Yeah. And for the conditional GAN that I'm working on, it's almost the same thing, but this time we are also plugging in another set of values, that is the populations.
yeah.
But I'm trying to make it work. It's not working yet. I'm hoping it will be done. Hopefully, like let's see. I have high hopes for that one because that was my target.
Yeah.
Like,
That would be good, right?
yes, one thing I got from yours, which is like you are not able to like identify like the time period for the embryo, right? Like you can't say like it is for the some stages like before this and this.
I can specify it. As you can see, the cells that are
Yeah.
generated.
So what you can do is like it's a little labor work. Like if you can do is like you can segregate the frames of the videos on the base of the labels that you have, like before this stage and this stage.
It's basically
Then
this GIF. Like you can see on this GIF, the population of A goes up. This is because this GIF has been like the photos are sorted based on the population of A. So
yes.
yeah, so
So
that is
it's like
it's like a brute force way, but I don't want to do that. I want to do something where the user can just enter the populations. It's possible, so let's just try. Why not? Otherwise, we always have this because I want to generate entire synthetic embryogenesis process. Right? We can just say that this embryogenesis is not real; it's synthetic and it's generated by a GAN. We can just one by one plug in those population values and generate 200 or so frames of a whole entire embryogenesis process that is generated by a model. Like from
yes, you can definitely do it. Yes, like during school.
but my main plan is that conditional model. But if that doesn't work, I'll go with this brute force approach that we just talked about. That's a good idea. I'll go with that.
Okay.
Yeah.
Yeah, sounds nice.
Yeah, it sounds pretty good. I also.
So it's I'm kind of deviating a bit, but anyways, I'm done with most of the work for GSoC. So I'm just doing this as an exploratory thing. I think this will help.
Yeah. Yeah, I think it'll be a good addition to, yeah. So I pointed out that a couple years ago we did the morphozoic paper. We did the morphozoic model, which is a cellular automata that it sort of like generates patterns and then classifies them. So it's not really like this, but it's kind of a similar type of thing where you have kind of generating. It's, I guess, like they're fake embryos. It's generating patterns, and then it's classifying them into different things, and it has to. So you have the cellular automata, and then you have this neural network. It's a standard neural network, and then it's classifying different patterns that are being generated as like an embryo or not an embryo or something else. That I like the way this works. This is of course the GAN. So we talked about that I think last fall in more detail.
The problem with this thing is that it takes a lot of time to train.
Yeah.
So on top of that, I have my exam, so I can't invest a lot of time. I'm sure this would improve within the next 2
Like
years.
Mike, I really want to I think we have done a lot of work and we should start making library work. So what is the schedule for the MSN exams? Like when they are
So exams, like it's ending on 30th. And
30th July.
yeah.
Okay.
And then
We
after
can,
that, I have one more Viva at third, but that's not a big deal.
so yeah, from next week onwards, I guess we can definitely start on building these libraries. I guess.
Libraries, you mean the backend for the website?
No, library means the Python. Like backend of the website is my work. Like I have to do that. The library we have to make together. Like yeah, converting this into a pep install version of DevoWorm.
Yeah, but I'm just setting up the code base right now. I'm still writing the like code base.
So what I'm saying is in the Pi elegance, we can start making library. We don't have to push it right away. We can start making a library, making all the requested functions like create file and help
Yeah,
bot and all these things,
I'm
and
not
we can keep
I'm
adding
not
these
into
things.
those things because this is the first time I'm doing
Yeah,
it.
yeah, I'm obviously here. Like, I'm going to help you out, and we are going to make it together. So I think from the next week onward, we can start doing this thing. In my
Yeah,
opinion.
sure. Like by this week, hopefully I'll be done with this generative model. I have to polish this one. This is not polished
Yeah,
because I used one of like those tricks that people use, like decaying learning rate and all those things. Like those things that I use on other models. I didn't get the chance to use it on this one. Like what you see here is just like a prototype. Like this is in fact a very bad pipeline for training. I have to improve it, and I'll obviously get a better model. But these models are kind of nuts. They're like 50, 60 MB each. Yeah.
Okay.
yeah, okay.
They're pretty heavy on the server. Yeah. I'm still trying to keep them as small as possible
Yeah.
so that it's not very heavy.
Okay.
Yeah.
Sounds good. Yeah. Thanks for the update. We'll keep. Yeah, we'll revisit this in coming weeks. I don't know. Well, we'll see how goes, and then eventually maybe we'll put this together. I assume this is going to go into the library and everything, so this will be
Yeah, the gang is already there in the library.
okay.
In fact, like if you clone it, you can even use it if you have Python and
Yes.
all the other utilities installed. You can even use it right now. But it's
We
kind
have to
of
just convert it into a pip version so that it will be easy installation. That's it. Like library is working right now.
this library has a lot of dependencies right now because the user has to also have Torch installed and all of those like NumPy, SciPy, OpenCV. Those libraries should also be installed to use this library because this is a very high level API. This is basically like a high level API. The easier it is to use, the more installations you need to do beforehand. Like
So what in the pip version we do is we usually create a file which is like requirement.txt kind of file where it has all the dependencies and whenever you are installing the DevoWorm AI library, it will install all the dependencies automatically if you don't have any.
it's about time you should start deciding for a name, I guess.
Yeah.
Good question.
For the you mean for the package or for the
For both, I guess.
Python package.
well, yeah, I guess we could think. Yeah, I mean we have the names that were sort of proposed at the beginning of the when you applied
Or
to
we
the projects. We don't have
we
to
could
use those. Yeah.
poll
We can,
make on Slack. Is it possible to make polls on Slack?
oh yeah, we can. I think you can do that.
Yeah, I think we should do that. Like it would be
Yes, yes.
to take their opinion as well because everyone is a part of it, right?
Yeah.
We can give like three options and we can like surely put a poll.
We can. Yeah, we
Yeah.
can also do it in Google Forms. I mean, if it's just a simple poll. That way, if people are outside of Slack, they can get access to it as well. But we can just propose some names and put them in sort of. We can just probably put them in public, I guess, and then we can put together a formal poll and then have people select and go on that official sort of response and see.
Let's see what we can do.
Yeah. My suggestion is DevoWorm AI. Yeah, I know we've you've kind of been putting that in the materials, and I like that. I like that term. I like that name. I think it's pretty good.
We could also do with
What?
we could also do with this one, deep worm,
Deep worm?
because it's deep learning and worm. So
Yeah.
Yes.
I'm just throwing things. I don't even know like anything.
Yeah.
You decide. It's up to you guys to decide. I'm just making. I'm just building it.
Okay.
I really hope this could pave the way for future projects. Like next year, if someone comes in,
Right. Yeah.
like there would be people doing GSoC next year. They could improve this even further and take it to another level.
And like,
Some more.
and like not only improving, like every year in GSoC, like there is a project for deep learn model training and machine learning project. So he can just make his project overall in a Python package and add it to it. So it will just getting more and more diverse and have more and more functions.
I think if we make this kind of a package, it would be like much more accessible to the people.
Yes, 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 yes.
Yeah.
That's
Like last week, remember, like as Susan said,
yeah, exactly. She said like it will be very useful for her.
Right. Yeah.
it's like generally people who are into biology are not like they're not really into Python, but they want to use those tools which are built on Python, right?
Right.
So this
Yes,
would
yes.
help them. That's like, which is why I'm really focusing on ease of use. Like it's not like I'm trying to keep it as easy and as minimal amount of code as possible, like lines of code.
Right.
It should work in like 4 lines of code, no more than that. Like like for every other, like for everything separately.
Right.
Yeah.
Yeah. Yeah. I like that idea. So yeah, we can send some candidate names out to see what people like, and then yeah, I think the usability is a good issue to focus on too. That being able to make it easy to use instead of even having that problem in one of my other groups where they don't like everyone thinks that it's easy to use, like some notebook that we're trying to use, and it's not as easy as we're running into some problems with it. So it's never a bad idea to make it as simple as possible. Just simply because it's sometimes it'll work, sometimes it won't. It's supposed to be universal, but I'm not sure about that. Anyways, so thank you for the updates. I wanted to mention that the second round of reviews are coming up this week for GSoC, so I'll be filling those out.
It's today.
Yeah, it starts
Yes.
today. So I mean, this is again, I'll just fill them out, and you're both passed. I mean, I don't think that comes as a great shock, but I will be suggesting some things. Like they always ask you, what could they improve on? So I'll think of something, and then we'll keep going on this in the coming weeks. So yeah. So okay, let's see. What do we have that I wanted to go through? Why don't we move on to maybe going over maybe some of the major tasks, like our issue board. So I think last week we talked about the GSoC issues, and I put those on the board. I don't know if people got those emails, but those issues are now on the board there. And so we'll go over the major tasks this week. Let's see if we have presenting now. Let's see. So can you see my screen?
Oh, yes.
All right. So we've got a lot of different things going on. Some of these are finished, been finished for quite a while. Some of these are ongoing. So the outline of action items for the periodicity paper that was done last week, but we're still working on that. So that's this. This is the repo here, and then the study plan is here. So this idea of creating this paper about periodicity in the embryo. So we've got division of labor here. If people are interested in a specific role, they can contact me. I didn't put down Susan. So, when she tells me what kind of specific role she wants to play, I'll put her in the list. Otherwise, it's just basic reviewing of the manuscript. But then, of course, if you want to do like some data science or some analysis or some other thing, then, some specific thing, we can talk about it. We have some data sets that we would use for this paper. We have zebrafish, a simulated embryo, and C. elegans. And then we have some ideas of how to compare them. So it's still ongoing. We haven't made any advances on that, but that's in progress. The axolotl embryo animations and segmentation is still in progress. Susan said she might send me some more data on that. But other than that, that's kind of been on hold. But I'm kind of having it in progress because I still kind of working on getting that together and getting it going. The Friday coffee hour hackathon that's been put on hold for a couple weeks. I've just been very busy, but we'll probably try to revitalize that soon. And again, this is just a meeting on Fridays that we can have where it's a more informal meeting where people can come in and share ideas or maybe work on something if they want feedback on it. It's just another chance to interact. We might even make this like bi-monthly or monthly. I haven't decided yet. So this is the periodicity paper. This is just kind of like the same issue, but just kind of the work on a more general issue. Complexity measures, we haven't talked about that in a while, but that's something that's involving Jesse was interested, and Dick might be interested, and of course George Mikalovsky. I haven't really contacted him about that yet, but a model of motile cells in embryo. I'm not really sure what that was. That might be the thing related to GSoC. Create embryo model in Blender. So this was something that Ujjwal was working on. Of course, this isn't like the priority during GSoC if he's doing GSoC stuff. So I didn't want to,
after like in the second or third week of August, I will start working on it again.
Yeah, just so it's in progress. We have some other things here. These papers is bacillary non-neuronal cognition paper. This was the one that we were working on with the bacillaria data, with the movement data on bacillaria. You have to revisit that at some point in the meetings. But that's still in progress. Krishna said he was going to present a paper. He hasn't done that yet. That's why I put it on hold. But he has done some stuff with tutorials, and he's created some tutorials for the Neuromatch Academy. My other group is working on Neuromatch Academy stuff, so he worked on some tutorials for that. Some coding tutorials and things like that. So those are actually, I didn't pull them up for the meeting today, but they're pretty interesting. That one, create Neuromatch tie-ins with DevoWorm. We didn't really do that. Neuromatch is this summer school that I've been working with the Neuromatch Academy on, and it's week 3 of 3. So we've been busy doing different things. We never really created a lot of tie-ins with it, but we might do that after the academy, just kind of with the contacts that we made there. So discuss AI innateness, that's on hold. Reorganize DevoZoo. This is something I think probably that we can put in finished, since we're kind of that's kind of a GSoC thing. Oh, it's not signed in. Okay. Well, we'll put number 7 in finished. when I'm logged in here. Okay. Axolotl montaging, that's something that is on hold because, when we start moving on this one issue on the axolotl stuff, then we move on this other one. So they're kind of linked. In the models for biological living networks, I'm not sure what that is, but that's on hold. Let's see. There's some other papers here that are, or ideas that are in the to-do list. I don't think any of these are really something that we are immediately able to do. We still have this Docker container for OpenWorm, the embryo visualization. That actually might be something, number 6, that we can take some of the stuff we've been doing in Devo or in Summer Code and contribute to. So I got an email from one of the people at OpenWorm. They're having their annual meeting in September, and he wants a 5-minute presentation from DevoWorm. So I'm going to go over some of the stuff we've been doing this year. And I'm probably going to do, of course, I'm going to do some of the deep learning stuff and then maybe put in some other things. I don't know, 5 minutes is a lot of time for, but it's, we'll probably do that. And then we also have this Docker container where all the other projects have contributed some sort of something you can run in the browser or something you can run on your hard drive that is like an illustration of that project. So some of the other projects, because they're based around actually just designing a software package, it's pretty easy for them. They say, okay, we'll just do the software package and you can run it in there and that's it. We don't really have a single software package associated here with the group, but I think maybe something with that's what I was, we were talking about the animation earlier. But it might be appropriate maybe to do something like one of the examples of the pre-trained models or something. I'm not really sure what's appropriate. I don't want it. It can't be something that takes a lot of processing. That's the problem. So that might actually not be appropriate for it. They've had some trouble with some of the simulations, like spitting out a bunch of files, output files, and filling in people's hard drives. So that's one of the problems they've had with these animations that they're running in this Docker container. So I don't know what the best solution is to this, but it's definitely something we should think about more. All right, let me go back and reshare my screen. There's one more thing I wanted to talk about on the board. There's this lecture on PCA, UMAP, and TSNE. So I've noticed that in a lot of the papers that you read on, not just in developmental biology, but in a lot of areas, they're doing anything with big data. They're doing something with these different methods. And so these are all basically dimensionality reduction methods. And in the case of UMAP and TSNE, they build these really beautiful visualizations of the data. The problem is it's very hard to interpret them. Especially with UMAP, they create these visualizations that are very hard to interpret. So I was thinking of doing a lecture on these different methods and kind of demystifying them. I've been doing lectures for my other group in conjunction with Neuromatch Academy, so then I'm getting good at the art of putting together like a half-hour presentation on something really complex. So problem is, of course, I don't really have any spare time to do that, but I'm planning on doing that soon. And then that's all I wanted to talk about the board. Again, if you have ideas for tasks, I think you can add to this board. Or if you want to email me something that we need to put on the board, let me know. again, these tasks are kind of at this level of granularity where, if we think about it, if we make some progress, it'll split into new tasks. But it's always good to have these sort of interface on a regular basis so that we remember. Good. So I think I'm going to do the last 20 minutes of the meeting. I'm going to talk about some papers in the reading queue. I haven't had a chance to do this in a while. There are a couple papers I have here. I'll start with this one, which is kind of a nice visual paper. Let me actually give you the link to the drive. So these papers, you can download them from Google Drive if you're interested. PDFs. All right, so the first one is this paper, Art and Science at the Cellular Mesoscale. And so this is, I think, a fairly new paper in Trends in Biochemical Sciences. And the thing about these trends papers is that they have these glossaries on the side. So they define different things in the paper that they mention. So that's always good if you're looking for, like, if you're getting introduced to an area and you want to know more about it. So this paper is a review. The abstract is experimental information from microscopy, structural biology, and bioinformatics may be integrated to build structural models of entire cells in molecular detail. These integrative modeling endeavors are a challenge in several ways. So one of them is the intrinsic complexity of biology. So you have models of many closely packed and heterogeneous components. So these are very realistic models. Last week we talked about OpenWorm being this hyper-realistic type of model. And that's kind of the same thing here is what they're focusing on. So, you don't have to build a model that's hyper-realistic, but a lot of the cellular models are, especially for like biochemistry. So they have these closely packed and heterogeneous components. The second issue is that the wealth of available experimental data is scattered among multiple resources and must be gathered, reconciled, and curated. So they're talking about different data sets that you're integrating and bringing together to do these experiments. I think you've seen with the data set that Mayukh is working with that there's this, even this problem in normalizing the data. But imagine doing this over like 3 or 4 different data sets. It's very difficult sometimes. And computational infrastructure is now only getting the capability of modeling and visualizing systems of this complexity. So again, we know this from like the deep learning venue where we're doing this work, and it's taking a lot of computational power and time, but you get these models that tell you, they're quite informative. So we present recent efforts to address these challenges, both with artistic approaches to depicting the cellular mesoscale and development and application of methods to build quantitative models. So they had an article, I think in 1991, so almost 30 years ago. They presented a feature in the same journal that created snapshots of the molecular structure of a bacterial cell by integrating the available information on ultrastructure, composition, and molecular structure. And so that is, I think, in figure 1. So this is in 1991. They were able to, they had these drawings of what the cellular mesoscale looked like. So they picked this object of measure, this level of measurement, and they visualized it back then. Nowadays, of course, you can do this computationally, and so this is what it looks like today. So they've gone from this picture to this model. And they define the cellular mesoscale as scale level bridging the nanometer scale of atomic structure and the micrometer scale of cellular ultrastructure. So they're talking about like basically molecular components like genes, DNA, RNA, things like that, and then moving up to this micrometer scale of the cell. So features on the surface of the cell, like receptors and other components. And as you can see, the cell is not just this round sphere, it's this very complex thing. And this is a cross-section of the cell. So that's the way they've kind of approached this. They've actually been able to use electron microscopy, so cryo-EM, and they're using this to image a lot of these samples that they're getting. They're also using game engines. So game engines are like software and hardware that allow you to do. So a lot of the simulations that they do in 3D, a lot of these types of simulations are done on game engines. And of course, we know game engines are used to play video games, but they're optimized for graphical processing. So that's how they use them. And of course, that's where GPUs come from. If you know anything about GPUs, NVIDIA was creating hardware for video games, and so this is kind of where this comes from. Integrative structural biology currently refers to studies where structural models of biomolecules and biomolecular assemblies are built based on a variety of data from multiple disciplines. So from subatomic imaging, EM or light microscopy, cross-linking constraints, proteomics information, etc. So this is where their different data sets are coming from. Quinary structure, interaction, often transient, of molecules into higher order assemblies, added to the traditional hierarchy of primary structure, sequence to quaternary structure, oligomerization of subunits. So this is where you're building from like just sequences of molecules to structures of proteins. So protein structures are actually pretty high dimensional structures. They have a lot of folding and a lot of attributes like that. And the way they've actually modeled protein folding is they use these type of game engines and other types of physics engines to model the dynamics of proteins. And protein
They
folding
use Unity.
What was that?
I think they use Unity because that's the most popular physics engine.
Yeah. They have some of these experimentalists will have their own different models for it, but they'll use things like basically like that.
Because Unity is also used in things like reinforcement learning.
Yeah.
Like they use plugins in Unity to simulate like spiders and other crawlers and try to make them walk and stuff. Like there are these famous from DeepMind, right?
Oh yeah, DeepMind. Yeah, they do a lot of that. Yeah, they use a lot of that sort of stuff too. But like protein folding actually occurs on a time scale that's pretty short, but it requires an immense amount of computational power because what's going on during that short time scale is a lot of like conformational folding and stuff. So you have this just an incredible amount of detail needs to be simulated. Finally, they mention virtual and augmented reality. And so why do they mention this? This is because you can use these technologies to visualize a lot of these models and do things like walk through the models and view them at different angles and pick them up and rotate them. This has been something that people have been doing for like maybe 20 years. And, I think in like some like gene sequence and protein folding simulations, they actually have used VR to a good extent to sort of visualize a lot of the higher order structure. Because as I said, in proteins you get a lot of folding, and that helps you to sort of, visualize what the problems are that needs to be solved. So they talk about the challenges of the mesoscale. They say it poses challenges that are testing the current limits of technology. This is because of the complexity of the mesoscale. Mesoscale environments are highly crowded, often with 20 to 30% of the space filled with macromolecular components that are highly heterogeneous and have flexible functional modes. So they talk about some data, some sources of data, and They talk about the state of modeling, which is pretty exploratory, so they don't really offer a lot of solutions. They just kind of talk about it as sort of a frontier. And then they talk about how do we expect progress to unfold. We also expect that progress will be driven by new ways of seeing, both in the experiments that probe the cellular mesoscale and in the visualization software we build to explore integrative models. In the early days of macromolecular structure, creative innovations in visualizations such as Linus Pauling's space-filling representations and Jane Richardson's ribbon diagrams revolutionized the way we think about biomolecules, crystallizing new modes of understanding. And so they expect there to be, this type of modeling to be driven by similar types of innovations. And of course, these are not really like hardware innovations. These are representational and sort of visual. So you have diagram systems, for example. Those are the types of things that they envision to be driving progress. And so I think that's true for a lot of types of modeling where we have a lot of data, but the harder part is like integrating it into a framework, and that's I think where you're getting there's a lot of opportunity there. So this is mesoscale illustrations as thinking tools. So this is an illustration of the cytoplasm to vacuole targeting process, autophagy on the left. Right is an illustration of the exosome created by Julia Jimenez during a graduate internship using cell paint. So there's a lot of opportunities for like artists and scientists to get together and do things that are to sort of build these models, these sort of visual models that aren't necessarily highly scientifically accurate. I mean, they're accurate to sort of the visual sense, but they don't have they're not quantitative models. But they give you ways to think about the system. So, to be able to see it like this, you can think about it in a different way than if you were just like reading numbers or if you were just talking in words. And so, so this kind of goes on about this. If you want to look at some good, nice images, both hand drawn and microscopy produced, then this is a nice paper to look at. They talk about drug discovery. They give some outstanding questions. So, what are the salient features and properties of the cellular mesoscale that impact the process of life? How can we target these properties in pharmaceutical interventions? What is the role of human interaction in integrating structural biology research? Another one is how do we create a comprehensive pipeline that integrates tools for data management and model generation? So, they're thinking about these questions that are somewhat similar to what we talk about in these meetings, but I wanted to bring them up in this context just to show how people are thinking about these different areas. Okay. So then the next paper is, I think I'll only talk about two today. I don't know if I want to talk about that one. Maybe this one. Okay, so this one is a developmental landscape of 3D cultural human pregastrulation embryos. And so this one focuses on our understanding of how human embryos develop before gastrulation, including spatial self-organization and cell type ontogeny, remains limited by available two-dimensional technological platforms. Here we report a three-dimensional blastocyst culture system. So what they're talking about is like cell culture, where cell culture is traditionally two-dimensional. You put cells on a plate and you plate them and they grow to, in populations, and then you can assess them with microscopy, and it's two-dimensional. But of course they have three-dimensional culture systems which require some depth. So sometimes they'll use like scaffolds to grow the cells on. Sometimes they'll just grow them in a three-dimensional chamber. And that gives you a little bit different perspective because the cells are allowed to move around in a three-dimensional way instead of just adhering to a surface. So they report on this blastocyst culture system that enables human blastocyst development up to the primitive stage. These 3D embryos mimic developmental landmarks and 3D architectures in vivo, including the embryonic disc, amnion, basement membrane, primate unique secondary yolk sac, formation of anterior and posterior polarity, and primitive streak. Using cellular transcriptome profiling, we delineate ontology and regulatory networks. So then underlying the segregation of epiblast, primitive endoderm, and trophoblast. So they're looking at like the molecular mechanisms for embryos divide into these three germ layers, and they're looking at the molecular markers of this as it emerges. So you can do this in 3 dimensions. You can't necessarily do this in 2 dimensions because you don't get those signals that you need from the 3-dimensional shape. If I put a bunch of cells in a 2-dimensional culture, I won't necessarily get that sort of organization. So then they do some implantation. Specific pathways and transcription factors trigger the differentiation of cytotrophoblasts, extravillous cytotrophoblasts, and syncytiotrophoblasts. And then they talk about that epiblasts undergo a transition to pluripotency upon implantation. And then they look at the transcriptomes of those cells. And together, findings from our 3D culture approach help us determine the molecular and morphogenetic developmental landscape that occurs during human embryogenesis. So they're looking at human embryos here, and they're building these 3-dimensional cultures. And so they're allowing them to form these massive cells that are 3-dimensional. And they're actually looking at gene expression in those 3-dimensional cultures. And so then they have, let's see. So this is the transcriptomic data. So these are the gene expression data that they use for these different genes. So they've assayed these different genes for these different conditions, and they have this is the general way how they assess this using heat maps. So you get they get fold expression differences is how you're measuring it. So they usually use some standard like gap pH, and you measure the activity of these different genes based on the housekeeping gene is what they call the standard, and then you have upregulation or downregulation. And so this is actually our scRNA-seq. So this is a little bit different method than just straightforward gene expression, but they get basically the same type of data. Having trouble moving this down. There we go. So they show some of the structure that you get in the 3-dimensional cultures. You get this anterior posterior polarity and formation of PSA. So there's some differentiation going on spatially. You get these different germ layers that form in these cultures. So here's a diagram of what's going on there. And they're using gene expression to assess, the formation of these different germ layers. So there are different genes associated with the germ layers, and they're looking at the patterns of gene expression there. So I think that's good for that paper. I don't know, it might be a little over the head of some of our computational people, but I think it's worth reading some of these papers from outside of your area of expertise because it's, informative. Dick says there is some literature on forcing embryos to develop in 2D. I mean, yeah, do you have a good sense of what that literature looks like, or is that just like a couple of papers here and there? So, yeah. Well, it's Well, I think people are, trying to explore 3D cultures, and now it's. I know there's been this work with embryoid bodies that people are really excited about, where they're actually trying to get grow like small neural networks out of a bunch of stem cells. So they're just growing these embryoid bodies, and they get them to differentiate into neuronal cells. And those have been they've had various success with that. They get these little balls of neurons basically that have electrical activity, but beyond that, they haven't really gotten any sort of like brain out of it. But I think that's interesting. See what people are doing with cell culture. Okay, everyone. All right. Thanks for meeting. See you later, Dick. Everyone else, have a good week. Talk to you on Slack.